Why is it more difficult to extract DNA from plant samples? Plant tissues often contain higher levels of carbohydrates, proteins, and secondary metabolites compared to animal samples. These substances can act as impurities during the nucleic acid extraction process, thereby reducing the overall extraction efficiency. Therefore, a different pre-treatment process is required compared to animal or bacterial samples, and in some cases, organic solvents such as phenol may be necessary. How can we simplify such a complex process? The EcoQprep Plant Genomic DNA Kit is a solution designed based on magnetic nanobead technology to consistently and conveniently extract high-quality DNA from various plant samples, such as leaves, roots, and seeds. DNA purification can be performed inside a clean bench using magnetic beads in a dedicated rack without the need for centrifugation, which helps prevent contamination caused by aerosols. Since it does not use toxic organic solvents like phenol, the kit ensures the safety of the user. It is easy to handle due to the absence of odors and hazardous substances, and waste disposal is also simplified. The extracted DNA can be used for various experiments such as PCR, gene cloning, and SNP analysis, helping to ensure reproducibility and convenience in your experiments. The EcoQprep kit is designed to extract DNA of consistent quality from various plant tissues. Experience the EcoQprep Plant Genomic DNA Kit, which allows you to easily obtain genomic DNA even from challenging plant samples.